right, ladies and gentlemen, we're going to start nucleic acids. Let's look at our examples. On the screen here, we have some meat with a bunch of fat on it, some eggs, some fish, and some fruits and vegetables. You'll notice all those things come from living things. All those things represent other organic molecules that we've studied. So the meat, remember, it contains protein and it also has lipids. The egg has an awful lot of protein, the fish has some protein, and the fruits and vegetables have a lot of carbohydrates. But you're also going to notice they all contain nucleic acids as well. So pretty much all of the examples of organic molecules that we've studied before are going to be representative examples of nucleic acids. In other words, your meat is going to contain nucleic acids, your eggs contain nucleic acids, your fish contains nucleic acids, and your fruits and vegetables contain nucleic acids. And that's because every living thing has DNA. And so whenever you eat any of these things, you are also eating DNA. So whether you're eating something that came from an animal, came from a plant, came from a fungus, or happens to invade you with a protozoan or a bacteria, they all contain DNA, and they all therefore contain nucleic acids. So let's get started and look at our objectives. What's the difference between RNA and DNA structure? Describe and draw the structure of nucleotides. Describe the function of hydrogen bonds and nitrogenous bases in nucleic acid structures. Explain the relationship between nucleotides and the DNA and RNA molecule. All right, so let's get started by looking at a little bit where is DNA and RNA located. So if we look at a cell underneath a microscope, and I'm sure you've looked at basically a cheek cell at some point in your studies, and you notice the center was that round dark circle. Well, that's because we can't really see too much into it because we didn't have very strong microscopes. But if we had a really strong microscope, we could actually look at it and find these little X-like structures that exist in the nucleus. That X-like structure is really tightly coiled and wrapped up on itself a DNA molecule. And then in the DNA molecule itself is made up of these special nucleotides. So when you think of DNA and RNA, it's RNA not so much, but DNA is going to be located in the nucleus of the cell, and it's just wrapped really tight along itself in the form of chromosomes. And if we unwrap those, it's kind of like taking a piece of yarn and unwrapping it until you get down to the last single strand, and that's when you'd have your DNA. So DNA is basically located in the nucleus, and it's tightly coiled up in something we're going to call chromosomes. So let's look at the function of DNA. And the function of DNA is going to be that it stores genetic information. And so this DNA, if you look over in this picture, it's just got a series of A's, T's, C's, and G's. And it's in this sequence of letters that's going to give the information that makes you, you. In other words, your hair color, your eye color, your body structure. All of those things come from the order in which these letters appear. And every single one of the cells that exists in your body has the exact same DNA, so all the cells have all of the information they need. And the important part about this, that this information gets transferred and passed on each time a new cell needs to be made, so that each individual cell has the instructions for this, so that everybody knows what can be made from one single cell. And like I could take an individual cell of yours and put it through replication, and then I could actually clone you, although there's rules and regulations against that today, but there have been cloning of cats and goats and certain animals. So that's why we also talk about the function of nucleic acids having the codes of life, or the instruction for building living things. It's also been referred to as the blueprints of all living things. So whether you're, again, an animal, a plant, a fungi, a protozoan, each of those contains the DNA with all the instructions necessary to build another cell. What are the elements that are involved in the nucleic acids? It would be carbon, hydrogen, nitrogen, oxygen, and phosphorus. The only one we're missing is sulfur, so it's CHNOP. The shape of nucleic acids is a helix, and whether it's a double or a single is going to determine whether it's RNA or DNA. And so a helix is kind of like a twisted string in a sense. I think of it as like a licorice twist. You know how they're all twisted and wrapped up against each other? So think of a helix as something that's twisted upon itself. The monomers that exist on nucleic acids are called a nucleotide. And this is a little bit more complicated than some of the other monomers that we've studied, and that's because none of the other monomers we studied had a lot of parts to them, in the sense that nucleotides have three distinct parts. They're going to have a nitrogen base. It means it's going to be contained up here. This will be the nitrogen base. Then they're going to have a sugar, and in this case, it's a 5-carbon sugar. And then they're going to have a phosphate group. And all three of these comprise of or make up a nucleotide. So if you look at this diagram here, the one you've got yellow, blue, and coming off of them is a different color with a letter attached to it. The letters are all your nitrogen bases, and your yellow and blue, one is a phosphate, one is a sugar. So again, it goes a phosphate, sugar, nitrogen base, phosphate, sugar, nitrogen base, phosphate, sugar, nitrogen base, phosphate, sugar, nitrogen base. So this molecule here has 1, 2, 3, 4, 5 nucleotides in a long chain. If we look at this diagram, again, you have your nitrogen base, your sugar, and your phosphate. Nitrogen, base, sugar, phosphate, nitrogen, sugar, phosphate, all the way on this side and all the way on that side. So you can see a nucleotide is made up of our three clear parts, the nitrogen base, the sugar, and the phosphate. Polymers, on the other hand, it's nice and easy because there's only two of them, DNA, or deoxyribonucleic acid, and RNA, which is ribonucleic acid. You want to be able to get to know those words, recognize those words, and say those words and spell those words. We're going to look at each one individually. If you look, here's a nice strand of deoxyribonucleic acid, and here's a strand of RNA, which is ribonucleic acid. So just looking at them quickly, you realize they're very similar, but they also have a lot of differences. So let's take a closer look. DNA is a double-stranded nucleic acid, which means it's made up of long chains of nucleotides wrapped around with two sides. Its nitrogen bases include adenine, thymine, guanine, cytosine. And from here on in, a lot of times you do need to know how to spell them and recognize them, but oftentimes we're going to just refer to them as A, T, C, and G because it's much easier that way. The sugar is deoxyribose, and that's really how the DNA molecule gets its name because it's deoxyribonucleic acid. So the sugars are going to determine the name of your polymers for nucleic acids, and then there's a phosphate group, and it doesn't change. It's going to be just the phosphate group. But if you look at this molecule, it's got very interesting makeup in that the nitrogen bases on either side of the molecule are going to be made from or connected by each other by hydrogen bonds. And if you remember earlier in the year when we were studying bonding, we studied covalent bonds, ionic bonds, and hydrogen bonds. And in particular, the hydrogen bond, we concentrated mainly on the water molecule in that you had your slightly negative O and your slightly positive H, and that's why it created such an interesting molecule. Well, here we have the same thing. We're going to have these two nitrogen bases are going to be linked together by a hydrogen bond. And if you remember, hydrogen bonds are the weakest of the three. They break easily. And that's how DNA is actually going to replicate itself. Those bonds are going to rip right open, and it's going to make copies of itself looking at either side. So just remember that the hydrogen bonding that's between the nitrogen bases is very important to the structure of the DNA because it makes it easy to break apart and easy to copy itself. If we look at RNA, RNA is single-stranded, made up of long chains of nucleotides. You can see it's still a helix. In other words, it's still twisted and made up of phosphate, sugar, nitrogen bases. But in this case, we have a difference in nitrogen bases. We still have adenine, but instead of thymine, there's uracil, and there's guanine and cytosine. So the RNA is single-stranded, and it also has a different nitrogen base. The sugar is also different. It is just ribose, hence its name. So the sugars are what give the nucleic acids their names. And in this case, RNA, it's ribose. So you can see that we have cytosine, guanine, adenine, and uracil as our nitrogen bases. And we'll talk about that a little bit later on how we use that information. But phosphate doesn't change. So there's basically some three differences here that we're pointing out between RNA and DNA. Double-stranded versus single-stranded. Nitrogen bases, you have three of them the same, but one of them is different. You have uracil in RNA and thymine in DNA. And then your sugars are different. You have deoxyribose in DNA and ribose in RNA. So why is DNA and RNA so important? And it's basically because of those letters, those nitrogen bases. They're going to contain the information that we need on how to make things. And it's also important because it carries these instructions that get passed on for the next generations. If the instructions don't get passed on to the new cell, then we have changes occur, and that can lead to all sorts of issues. But let's look at something hypothetical here. If you look at the sequence of A's, T's, C's, and G's in here, let's just pretend if this was, I was reading some of the DNA and I had this sequence, it could possibly make brown hair. Now it doesn't because I just made it up. But if it did, if we look at what's the sequence, the difference for making black hair, you'd see there's only one letter difference. And that sequence, again, because you have a different nitrogen base, it becomes in a different order, can create something different. In other words, your instructions are different. So look at the next one. Here, you still have A, and then you have a T, a one letter difference, and then you have two more differences. So you have slight differences between each, which is going to give you a different characteristics. So the order in which the nitrogen bases come in determines the genetics of that individual, or in any case, an organism. So it is important, and it is important because they get transferred each time that a new cell is made, and then a new organism is made. We also have to remember that when we talk about nucleic acids, the same principles of dehydration and hydrolysis apply. So if I wanted to make a whole bunch of DNA, I would take my nucleotide and an enzyme and then add it in, and I can create my DNA molecule. Again, you're going to make sure that enzymes are involved, and you are going to make water as a result of putting the nucleotides together to form your DNA and RNA. The opposite, if I have a big strand of DNA and RNA and I want to break it down, then remember I'm also going to add in an enzyme, add in some water, and I can then break down my DNA and RNA into the complete sets of nucleotides. So you do need to know that these two reactions do also apply to our nucleic acids. That about does it for our nucleic acids. Remember, can you give me an example of where nucleic acids are found? What is their function, the shape, anything you need to know, and everything you need to know about the monomers and the polymers? Why is it important, the sequence of the nitrogen bases? How does hydrogen bonding play an important role in DNA and RNA? And what's the differences between DNA and RNA? And do you understand dehydration reaction and hydrolysis as far as nucleic acids are concerned? That about does it. See you in class.